Okay, great. Thank you very much to Crescio and to Thilo for inviting me to take part in this session and in this conference. So what I'm very excited to tell you about today is some recent work that we have done on understanding how non-random structure emerges in recurrently connected networks from a particular spike timing dependent plasticity rule that depends on triplet interactions of spikes. So we know that Hebbian plasticity is one of the best known activity-dependent mechanisms that connect cells that fire together. So we know that the persistent firing of neuron A, if that drives the persistent firing of neuron B, then the connection between A and B will be potentiated. And this kind of Hebbian plasticity has been sort of argued to be one of the key mechanisms for the emergence of cell assemblies that have also been termed Hebbian assemblies. So these are groups of neurons that are strongly connected to each other. And they become strongly connected to each other because they're repeatedly co-activated. So they experience correlated activity and they become strongly connected. And these cell assemblies are relevant because when they're present in recurrent networks, such as the ones I'm showing here on the slide, they generate very different types of dynamics than these networks that just have uniform and random connectivity throughout. So here we see the activity of such a uniformly connected network at the top, which is asynchronous and irregular. And at the bottom, we see the activity of these structured networks that have these cell assemblies in them with higher probability of connection within an assembly or stronger connection weight. So these have been suggested to be the building blocks of cortical microcircuits and to be recruited in the execution of different types of functions. And what is interesting about them is that they enable the cortex to generate these reverberating patterns of non-random activity. These have been implicated to be responsible for the representation of different stimulus features. For example, different tastes in gustatory cortex, and more prominently in the visual cortex, various studies have shown experimentally that such cell assemblies indeed exist. So here I just show an example from the work of Tomašić-Flogel, where they measured specifically connection probability for neurons as a function of their similarity of stimulus preference. So these are simple cells in the visual cortex that are tuned to different orientations, as is shown here in the schematic. And if one looks at the probability of connection, you see that neurons that have similar stimulus preference are more likely to be connected than neurons that have orthogonal stimulus preference. And what is interesting is that this type of pattern of connectivity, presumably giving rise to these self-assemblies, emerges after eye opening. So in the mouse, eye opening is around P15. And so it has been proposed that potentially this structured visual input coming through the eyes after eye opening could give rise to the formation of these cell assemblies. Now, cell assemblies have also been seen in other kinds of brains and brain circuits. So here we see an example of the optic tectum in the zebrafish. This is functional imaging, so 2-photon calcium imaging at different ages. So DPF stands for days post-fertilization. What is important here is the emergence of these beautiful connectivity structures. So this is all functional connectivity, so functional cell assemblies in the optic tecta of these animals. But unlike the visual cortex example that I showed you on the previous slide, here it seems that these types of cell assemblies emerge completely in the absence of patterned external input. So you can block visual input by removing the eyes, and you can still see, or experimentally still see, the emergence of these functional cell assemblies. So this is something that puzzled us. And when we looked at the literature, especially in theoretical models of how these cell assemblies emerge, we saw a lot of studies that focus on this patterned external input that gives rise to these assemblies. But we know experimentally, at least from this example, that they really can come about in the absence of patterned input. So we asked what could drive the emergence of these self-connected assemblies in the absence of patterned input. And our hypothesis was that this is driven by internally generated network interactions. So what I will describe to you now is some work that we did that was primarily led by my postdoc, Lisandro Montangi, and my PhD student, Christoph Miel, that was published recently, that tried to answer this question. And when we started to look at recurrent networks and ask how could the emergence of these assemblies come about, we looked at sort of popular models of synaptic plasticity. And so one of these models of synaptic plasticity is spike timing dependent plasticity. So this is effectively the spike-based implementation of this popular Hebbian activity dependent plasticity that I described at the beginning. And so, I mean, I probably don't need to explain this, but according to this pairwise STDP rule, a synaptic connection between a presynaptic neuron J and a postsynaptic neuron I will undergo potentiation or LTP upon persistent presentation of a pre followed by a postsynaptic spike, and vice versa. If the postsynaptic spike precedes the presynaptic spike, then the synaptic connection will undergo LTD. So this was originally measured by B and Pu in '98 and has really been the subject of a large study of theoretical work that looks at what is the role of this particular plasticity rule in shaping non-random organization in these recurrent networks. However, if we think about how this pairwise STDP rule acts, if we have this one neuron A that persistently drives the firing of neuron B, because of this acausal or asymmetric structure of the pairwise STDP rule, it's not hard to imagine that this rule will always actually prefer unidirectional connections between two neurons. So you will always only potentiate the connection from A to B, but not the connection from B to A. So this rule is not able to generate these bidirectional connections, and these bidirectional connections are important for the generation of these cell-connected assemblies where you have many, many neurons that are connected to each other. And indeed, us and others have shown that this rule, indeed, when you put it in recurrent networks, cannot on its own generate these self-connected assemblies. But more recently, and this includes some work that I have done together with Jean-Pascal Pfester, is to consider that perhaps pairs of spikes are not the main building blocks for synaptic plasticity, but maybe the next contribution coming from triplets of spikes is important for driving plasticity. So this type of rule is called a triplet spike timing dependent plasticity rule because it relies on evoking potentiation or depression depending on the timing of triplets of spikes. So potentiation is induced by having two postsynaptic and one presynaptic spike. And so basically this third spike, which is effectively the second spike in the postsynaptic neuron, what it does is it modulates the amount of classical LTP that you will get from this classical pairwise STDP rule. And similarly, you can have a third spike, which effectively is the second presynaptic spike, which will modulate the amount of LTD or depression that you would get in classical pairwise STDP, the STDP rule. So this rule was proposed by Jean-Pascal, and it was shown to capture a larger amount of experimental data than the pair-based rule, and especially these experiments wherein they induce plasticity by pairing pre-post spikes multiple times, but where they vary the frequency with which these pre-post spikes are presented. So you can have a scenario where you do pre-post, you wait for a long time, you do pre-post again, or you can have a scenario where you do pre-post at a very high frequency. And so these kinds of experiments can be much, much better described by this triplet rule. And so based on some preliminary work that we had done at the time, we postulated that this type of plasticity rule that relies on these triplet interactions might be capable of driving the emergence of these self-connected assemblies in these recurrent networks. And so this is what we started to do in this work. We consider the recurrent network of excited tree neurons, indicated here by the triangle. So these could be, for instance, the pyramidal neurons in the cortex. The connectivity in this case is all-to-all, but this is not that important. It could also be sparse and random. And these, we describe the activity of these neurons with inhomogeneous Poisson processes. So as we know, this is an appropriate representation if we study even, say, leaky integrated fire neurons that fire synchronously. But what is important for us is we use this framework so that we can calculate a lot of the contributions that emerge through the plasticity rule for the plasticity of the synaptic connections. So we use these Poisson neurons for convenience. And we drove this network with external input, but I should point out that this external input was completely unstructured. And so What I will show you in a moment is how these synaptic connections self-organize in a way that they build or organize into a non-random structure purely from internally driven correlations. We also had inhibition in the network, and this inhibition was not plastic, and it was just there, was always scaled to excitation just to prevent activity from blowing up effectively. So this, I should point out that mathematically we can treat this network using the framework of Hawkes processes, which are mutually exciting point processes. So what we can do is we can write the average rate of change in the synaptic strength between presynaptic neuron J and postsynaptic neuron I under the STDP plasticity rule. And we can do this by effectively writing the change in the weights as the product of the time-dependent cumulants of different orders. So these are these C's and K's that I will explain in detail in a moment, and the STDP functions. So the STDP functions here are denoted with the L functions. So L2 corresponds to the pair STDP rule, and L3 corresponds to the triplet STDP rule. And I have written here the equation in the most general form, where we explicitly write the contribution of all cumulants of second and third order as they combine with these two plasticity rules that use either pairs or triplets of spikes to induce potentiation or depression. So let's look individually at each of the terms that contribute to plasticity in this framework. We have two types of second order cumulants. We have first the pairwise cross correlations. So these are this is effectively the probability of joint spiking of the pre and the postsynaptic neuron as a function of the timing delay between the two neurons. And so as you see, the plasticity here, the contribution of this particular pairwise cross correlation to the plasticity comes both through the pair and through the triplet STDP rule. So it is integrated together with the two plasticity rules. But we also have another second order cumulant, and that is the pairwise autocovariance, the CII term, that only contributes terms in plasticity when it is combined with the triplet rule. So it is not present in the term with just the pair STDP rule. So this is a novel term that we bring completely from scratch in our analysis. And another term that we have, because we consider this triplet spike time independent plasticity rule because of the contribution from triplets of spikes, is this third order cumulant that I have denoted here in purple with Kij. And unsurprisingly, this term only appears in the second and third line of the equation here because it only contributes to plasticity as it is combined with the triplet STDP rule. So this is the equation for the weight dynamics in the most general form. I should just point out that due to other studies by Jean-Pascal, they actually showed that a more hybrid version of STDP, so one that only uses triplets of spikes for potentiation and pairs of spikes for depression, is just as good as capturing the weight dynamics in various experimental studies as the full triplet model that also uses triplets of spikes for depression. And so from now on, for simplicity, I'm going to be using this so-called minimal triplet model where, as I said before, triplets of spikes are only used for potentiation. So this allows us to slightly simplify the expression that we have for the overall weight dynamics by effectively eliminating this last line, which is the contribution of these triplets to long-term depression. So what can we do with this equation? So notice here, I have grouped the second and the terms in the second and first line according to the rules. So the first line is the contribution of the covariances to the pair rule, and the second line is the contribution of these cumulants to the triplet rule. But we can actually rewrite it so that we group it not according to the contribution to the rules, but according to the contribution of these different cumulants. So now each of the three lines of this equation denotes the contributions of either independent spikes, so completely independent of joint firing statistics such as correlations in the first line. The second line denotes the contributions of all these second order cumulants, so the cross correlations and the autocorrelations. And the last line gives us the contribution of the novel terms that are purely used by the triplet rule. So these are the autocorrelations and the third order cumulant. Okay, and I should point out that we make a simplifying assumption where we set this first term to be equal to 0 because we're only interested in how these internally generated correlations, namely the pairwise or these triplet correlations in the network, so completely intrinsically generated by the dynamics of the network, drive plasticity. So we are not interested in a regime where changes in firing rates dominate plasticity. So that's why we set this first term to be equal to 0. Okay, so what we can do now is we can actually rewrite this covariance term in this equation. And here I've only illustrated the pairwise covariance terms, so only the first expression in this equation. We can rewrite these covariance terms in terms of what we call structural motifs. So by doing this, we can actually break down these second and also third order cumulants into expressions that include the contribution of every spike that's propagated in the network through existing synaptic connections. So taking into account the full recurrent connectivity in the network. So we can take this cross covariance term, for example. So this is the expression indicated here with the asterisk. And we can write it by summing over all the possible paths from every possible source neuron K. So here we sum over the neurons K, and then we sum over all the possible paths that a spike takes from the source neuron K to the pre and post synaptic neurons I and J. And this is perhaps better illustrated by looking at this schematic. So we can write this Cij, this pairwise cross covariance shown here in this line of the equation, as the sum over all the spikes that go from the source neuron K and go either to the postsynaptic neuron I or the presynaptic neuron J via these two different paths that we call the alpha path and the beta path. And so I should point out that this framework was originally proposed by Ravi Tenenbaum and Burak for only the pair-based rule. And what we have done here is we have extended it to include terms also for the triplet STDP rule. So we have this now expansion, and we can do this also for this term and also for this other term. So for all the contributing cumulants of different orders. And what is interesting here is that we get these by rewriting the change in the synaptic weights in this way. We get an expression that depends on these so-called motif coefficients, which are functions, as you could see here, of the spike timing dependent plasticity rule, of the length of the alpha and the beta paths, and also of another variable that I haven't mentioned so far, but that is also important and is indicated here by this variable E, and that is the evoked postsynaptic current, or the EPSC. So it's the effect that a given presynaptic spike has on the sort of probability of spiking of the postsynaptic neuron. So we can go from this kind of equation into an equation where each of these terms can be rewritten by using these motif, structural motifs, together with the motif coefficients that tell us how strongly each of these structural motifs contributes to the weight dynamics. So let me give you some examples. So what I show you here are just some structural motifs under the pair-based rule that emerge when you decompose this pairwise cross covariance into these structural motifs. And so these are not new. These were previously proposed by Ravi Tenenbaum and Burak, but I just want to give you the intuition. So one interesting motif is this so-called 1-1 motif, where basically the alpha and the beta path have length 1. So here, always J is presynaptic and I is the postsynaptic neuron. And the reason that this is a 1-1 motif is because we have a source neuron K, indicated here in cyan, that projects with a single connection to the presynaptic neuron, and hence beta is 1, or to the postsynaptic neuron, and hence alpha is equal to 1. But we can also have other motifs where, for example, the source neuron is exactly the presynaptic neuron. So that's why here the length of the beta path is 0, because there's basically no connection onto itself. But there's a single connection from the presynaptic neuron to the postsynaptic neuron. That's why alpha is equal to 1. Or we can have a different motif where now the postsynaptic neuron is the source neuron. So here the alpha path is 0, but the beta path is 1, because we have this path that can influence this connection from the postsynaptic to the presynaptic neuron. So I hope this is at least intuitive. Yes. 5. Okay, good. So I hope this is intuitive, at least for how to rewrite these contributions. for the pairwise cross covariances, but let's actually look at what are some of the novel structural motifs that emerge under the triplet STDP rule. So what we have here is a way to rewrite the new term that emerges under the triplet rule. So here we have the triplet rule as L3. So we can rewrite this pairwise autocovariance again as a sum of these structural motifs. And what is interesting here is, in addition to the standard alpha path where we influence the neuron by looking at the influence of spikes from all other source neurons in the network, we have basically an identical path from the same neuron, source neuron, to the same postsynaptic neuron, but now via a delay. So this is now the contribution. So by introducing these triplets of spikes and by allowing to kind of take into account more than just a single postsynaptic spike in the induction of plasticity, we can now include paths that sort of structurally take the same path through the network. They go through the same neurons, through the same connections, but with a given delay. And so this is a new novel term that, as we show in a moment, plays an important role in determining what happens in the network. So what I will not show you now, but we go into detail about this in the paper, is how to also decompose these third order cumulants in the same way using these structural motifs. So let me just give you some examples of these motifs. When we introduce the gamma path. So here again, we have a presynaptic neuron J and a postsynaptic neuron I, and we have a source neuron K. So as before, we can go into the presynaptic neuron via a single path. So that's why beta is equal to 1. But we can now go to the postsynaptic neuron sort of via two connections, a direct one, which is the alpha equal to 1, and a delayed one, which is the gamma equal to 1. We can also look at two other interesting motifs that come in this framework that for sure do not affect the pairwise covariance, and therefore we know do not play a role in the induction of plasticity under the pair-based rule. And these are very interesting because they completely do not affect this presynaptic neuron. So these for sure cannot exist in the case when we study the pairwise STDP rule. So here, either the source neuron is either the postsynaptic neuron, so we kind of have this interesting loop that goes back to some source neuron in the network and comes back to the postsynaptic neuron again. Or we have this other scenario where we have a source neuron that again projects with this delay into the postsynaptic neuron. This particular motif that I showed you here, where we have the postsynaptic neuron to also be the source neuron that has these interesting loops, we call them loop motifs, is actually very important for the formation of these assemblies. So I'm just gonna flash this very briefly to show you that what we have achieved here is we have found a way to decompose completely the equation for the induction of synaptic plasticity, where we had the contribution of all these covariances, so the second order cumulants and the third order cumulant, as effectively a linear combination of these structural motifs. And so these are just the examples. You can study them. We can write them exhaustively for the case that we consider. The really interesting ones, however, are the ones in the second and the third row, because these are completely novel structural motifs that emerge under the triplet STDP rule. And especially interesting are the ones that I have circled here in gray, because these are the ones that we call loop motifs that I will show you in a moment give rise to the emergence of these assemblies. Okay, so how are we going to study the emergence of these assemblies? We're going to look at the triplet rule again. So we have the potentiation part, which is evoked by triplets of spikes. And we have the depression part, where we're going to modulate the time scale of, or effectively the influence of depression by broadening this influence through the time scale of depression. And this has been motivated biologically in various experiments where, for example, the application of different types of neuromodulators can indeed deepen this influence of this depression. And so we can look at the evolution of these different types of motifs that we have in the network. And so here we have the motif coefficients as a function of how deep this depression is in our plasticity rule. So classically, people think that this common input motif, this 1-1 motif shown here in the green, is the one that actually drives the emergence of assemblies. But what we find in our work is that although this motif is important, you see here, it's not the dominant motif. What ends up driving the formation of assemblies, self-connected assemblies in our work, as we show here at the bottom, as we vary this depression strength of the plasticity rule, is the relative interplay of two types of motifs. We call them feed-forward versus reciprocal motifs. So the feed-forward motifs are ones where the alpha path, so this is the one that goes into the postsynaptic neuron, is longer than the beta path. So here with this blue ellipse, I'm trying to indicate all the feed-forward motifs in our framework. But we also have these reciprocal motifs. And actually what we see at the bottom is as we vary this depression strength of the plasticity rule, we see that these reciprocal motifs, this is basically a zoom in on what really happens in this plot. We see that these reciprocal motifs cross from being negative to being positive right around the values of this depression parameter eta minus, where we see the emergence of these assemblies. And so when these reciprocal motifs are negative, assemblies cannot form. So these are the cases for very weak depression in the network. But as these reciprocal motifs turn positive, then they can effectively support these bidirectional connections that are important for the emergence of these assemblies together with the feed-forward motifs. Okay, so I just want to summarize the emergence of these assemblies using a graph theoretic measure known as the clustering coefficient, which tells us here exactly how these, or quantifies how strong or how tight these assemblies are as a function of this modulation parameter. And I'm going to end by really stressing the importance of the novel terms that we have derived under the triplet STDP rule that are important for the emergence of these assemblies in contrast to the pair rule. So if we only consider these motifs shown here in the top row that also take part in plasticity under the pair rule, we basically see very weaker or not as strong clustering shown here with the blue curve when we apply it in our plasticity framework. What we need to do is include some of these terms that emerge uniquely under the triplet rule, and especially these so-called loop motifs. So here I include the terms that participate in plasticity with the pair rule, and only these two novel terms that we call loop motifs. And if we compare the performance with just these motifs, we see much weaker clustering in these self-connected assemblies. But if in addition we add these loop motifs, we see much better clustering in these self-connected assemblies, even better than actually when we include all the terms in the network. Okay, and one can actually visualize what this connectivity looks like as a function, if one looks at these connectivity matrices as a function of this modulation parameter in plasticity. Okay, so I'd just like to summarize with what I have shown you. I sort of divided my talk in two parts. In the first part, I explained to you how we managed to derive a framework that gives rise to plasticity in recurrent networks that includes second and third order correlations. So we utilize not just the pair STDP rule, but the triplet STDP rule that utilizes the contributions from these third order correlations. We showed that we can rewrite these contributions from the cumulants through these structural motifs, and that we showed which particular structural motifs that are novel under the triplet STDP rule are important for the generation of these self-connected assemblies. And at the very end, I just want to thank the people who did this work. This was my postdoc, Lisandro Montangi, who did an incredible work of plowing through these calculations together with my PhD student, Christoph. And I'm happy to take questions in the remaining 2 minutes. Thank you.
Thank you. Very interesting presentation. So I will go and turn the first comment into a question. That's from Mark van Rossum. This is Song and Abbott in 01 show that recurrent connections can arise with pairwise STDP as long as the time constants for LTP and LTP are different. So the question in more general would be, can you get assemblies with pairwise rules?
So the question was that Song and Abbott showed that you can get bidirectional connections under the pair rule if what is the case if you have?
So the
time constants
LTP
are different.
and LTD are different, right?
So
it's a comment, but I'll turn it into a question. So yeah.
No. So as we show here, if you only use the pair rule, let's look at this here. If we only use the pair rule, and if we look at the contribution from only these pairwise cross covariances, we cannot actually get these self-connected assemblies. Here, indeed, we are increasing the dominance of depression. That's what we're doing by this depression parameter eta minus, and we cannot get as good assemblies that look as tight as what we can get with the triplet rule. I mean, so Ravid, Tenenbaum, and Burak argued that you can get assemblies with the pair rule, but they had to assume two things. They had to assume a symmetric STDP rule, so they had to basically eliminate this causal or this asymmetric property of the STDP rule, and they had to introduce synaptic delays. So they had to do these two tricks in order to get assemblies with the pair rule. But as we show here, you cannot get and in that case, I should point out that when you do get these assemblies with the pair rule, you get them by amplifying this so-called this common motif, so this 1-1 motif. The way we get assemblies here is very different. Here we get assemblies by promoting what I call these feed-forward motifs over the reciprocal motifs. And so this is interesting because it's a different way to get assemblies, and it's also interesting because the assemblies look different. If you just promote this common input motif, you get perfect assemblies where one assembly is self-connected within each other and completely disconnected from the other assemblies. But by doing this in our way with these feed-forward and reciprocal motifs, you can get assemblies that are perhaps more biologically realistic because they're not decoupled from each other.
Right, thank you. To keep on time, we'll go to